Welcome to the 54th InterScience Conference on Antimicrobial Agents and Chemotherapy, or ICAAC, and this is ASM Live. I'm Michael Schmidt from This Week in Microbiology, and together with the program committee's help, we've selected some of the science that's being presented here at this meeting and asked the authors to come and discuss this work. This morning we're joined with two of the scientists, Dr. Bruce Geller from Oregon State University and Dr. David Greenberg from the University of Texas at Southwestern. Together they're going to share with us this new and exciting development on new antimicrobial strategies that silence NDM, or the New Delhi metallo-beta-lactamases. Those of us who pick up the morning paper each day in our hometowns appreciate that NDM is something you don't want in your hospitals and you don't certainly want to catch if you're a patient. So these gentlemen have been pioneering a really cool technique where they've created a synthetic DNA molecule that can bind and silence the gene responsible to cleave all these beta-lactam drugs, or it's a very effective beta-lactamase inhibitor. And this is, of course, what everyone is concerned about as we just listen to the CDC and they talk about CRE, or the carbapenemase-resistant Enterobacteriaceae. We talk about KPC, or the Klebsiellas. All of these things, or the more important classes, the ESBLs. And NDM is just but one component. So gentlemen, thank you for joining us here at ASM Live, and I'm just going to start with Dr. Geller. Why don't you share with us the results that you're going to present in your poster at 11 o'clock this morning. Dr. Geller.
Thank you. Thanks for the invitation to be here today. It's my pleasure. So we've been developing this new type of a compound that's an inhibitor of the carbapentamases, as you pointed out. And basically the results that we'll be presenting at the poster show that this compound actually reduces the MIC, the minimal inhibitory concentration of meropenem when it's given concomitantly to the bacteria. Not only does it reduce the MIC value of meropenem and restore susceptibility to meropenem-resistant bacteria, but it also kills the bacteria. We have at least a 3-log reduction in the viability of the cells when given this compound in combination with meropenem. And we know how important it is when prescribing antibiotics that if you have an opportunity to select a cidal or drug that kills bacteria, how much more effective they are in terms of restoring the health to the patient than the bacteriostatic classes of antibiotics, where you have to use those drugs in concert with the patient's immune system to clear the infection. Yes, that's right. And of course it's the meropenem that's doing the killing, but it's our compound that we've developed that's actually lowering the resistance of these bacteria to the meropenem and restoring these pathogens to susceptibility. So, Dr. Greenberg, what's your role in this exciting process?
Well, thanks for having me. We've been working on using this platform in a number of different ways and in a number of different bugs. And this really is an antisense approach, where instead of developing things that could be very broad spectrum, we try to silence pathogens in a gene-specific way. And there are different ways that you can do that. You could target antibiotic resistance, which is what we're talking about today, modulating resistance so you can restore resistant organisms to sensitive ones. And you could also use oligos to target pathogens the way most antibiotics work, which would be targeting core essential features of the bacterial pathogen. And one of the co-posters takes that approach at 11. And then the third pathway that we think about using antisense would be modulating virulence itself. And so those are 3 different roads or paths that we're using to explore antisense as true anti-infectives. And NDM would be an example of modulating resistance, as we've talked about. So we work together in all phases of these studies and have looked across different pathogens in different ways using Antisense.
So in essence, to reduce it to 2 words, is you're developing magic bullets.
Right. So I'm an ID physician, and this meeting is full of examples of how we've ended up in the crisis that we're in terms of antibiotic resistance, and I think a lot of people are aware of the different ways that we've come to this point. One is the indiscriminate use of antibiotics, and the use of antibiotics and all the collateral damage and all the other organisms that we affect when we're potentially treating a patient. So our view is if we can come up with situations where we can target therapy in a very directed way, that could have the benefit of preventing some of the collateral damage that we see and not having off-target effects and either affecting our own microbial flora and really focus on a specific pathogen.
That's what everybody has been hearing more and more about. At the general meeting of the society that occurs in the spring, we saw paper after paper on the importance of the microbiome, and what was absent from many of those discussions are the consequences associated with the use of broad-spectrum antibiotics. So I think hopefully we'll see a renaissance of new anti-infectives that are specific for, as you said, particular genes, particular pathogens, particular virulence pathways. And so I'm really excited about this family of papers that are coming out at this year's ICAAC. Now the hard questions. How do you deliver these DNA molecules? Is it IV? Is it intramuscular? Is it a pill form? When do you hit? So it's the whole combination of when do you deliver it? How long are they in the patient? Do you have to constantly infuse them? All of those tough questions.
Well, the specifics we haven't gotten into.
You're still figuring out.
It's all in preclinical at this point, and we've yet to address some of those questions. Certainly, we've thought about those things. But I would like to point out that we did publish a paper last year about this time that showed in a mouse model of pneumonia caused by Acinetobacter baumannii that these oligomers targeted to essential genes could reduce inflammation, reduce the viability in the lungs, and of multi-drug resistant Acinetobacter. So that study showed for the first time that we could deliver it to the lungs directly instead of IV, for instance. So that was one of the significant findings of that particular study that we both co-authored.
These molecules are stable in serum. We could envision delivery a lot of different ways, and there'll be some examples today in the posters of different ways that you might think about it. As an example, if you were giving targeted therapy for Pseudomonas or Burkholderia, that's something that I work on in the lab. We're starting to aerosolize these or nebulize these for targeted organ delivery. So they could be used in a targeted fashion or potentially used in an IV fashion as well. So I think the answer is sort of yes to all of the above. We could envision different ways to deliver it for different hosts or different situations, clinical situations.
So this is an absolutely fascinating area of truly gene therapy for bacteria.
This is, I sort of think of this as the siRNA version of prokaryotes, because really fundamentally what we're doing is targeting a gene, blocking translation and production of protein of that gene product. So if you have a genome of an organism, theoretically you could now design these to target whatever gene of interest you were going after.
So this is one of those cases where we're really fulfilling the title of this conference. It's really interscience, because you have to understand your microbe, so you have to have the basic scientists. You have to understand the pathogenic process in the host. So this is where the clinical medicine comes into play. You have to understand the pharmacodynamics, and so here's where our friends from pharmacology come into play, and then you have to have all the associated issues of stability. And it's great that we have conferences like this where you can kibitz with your colleagues as you're just sitting having coffee.
Yes, indeed. I'd like to point out that I think while the two of us are sitting up here, there's a lot of people that are working in the background and have given us great support and work as partners with us. It's a perfect example of the partnership that was described by Dr. John Rex in his keynote address on Saturday. That is, we have a partnership not only between the two academic institutions that we work for, but we also work with closely with Sarepta Therapeutics, which is a company that makes these wonderful compounds. And of course, we're supported by the NIH in research grants. So academics, industry, and government participation in development of this new technology.
So why don't you take us through the key findings of your paper, and then we'll hopefully take questions from our audience here in the room and also from the internet. So what are the key findings that your posters are going to present?
Should I start out on the NDM-1 poster? And yeah, so I outlined the basic results of this compound that's targeted to the NDM-1 gene, and it's basically pretty simple. We give this compound, put it into culture with either Klebsiella, E. coli, or Acinetobacter that are expressing the MDM-1 carbapenemase. And we then measure the MIC of meropenem at various concentrations of our compound. And what we're finding is that we reduce the resistance from a level that's very high to within therapeutic range. For instance, in Acinetobacter baumannii, the MIC in the strains that we've looked at, the MIC for meropenem is something like 256 micrograms per mil, which is way out of therapeutic range. We can reduce that down to about 8 micrograms per mil using 8 micromolar of our compound.
Well, that's not much at all.
Yeah. So, and so we've shown that we can reduce the MIC. That was the first step. The other thing we looked at then was to take samples from these cultures in vitro and do viability counts. And we've shown that the viability of these bacteria in the presence of our compound, meropenem, is reduced at least 3 orders of magnitude, and I think in some cases by as many as 5 orders of magnitude. And then there's a third part to the presentation poster where we actually looked at some of a biochemical approach to looking at the reduction of the carbapenemase in the periplasms of these 3 pathogens that were treated with the drug. And what we found was that the carbapenemase activity in the periplasm was inversely proportional to the dose of our drug that was given.
So really exciting demonstration of the utility of this approach, and it's very encouraging. Dr. Greenberg, are you on a companion poster, or?
Well, we have 3 posters today, the NDM-1 story, and actually all 3 posters complement each other in the sense that I think they really outline some of the 3 major pathways that we could think about using antisense, modulating antibiotic resistance. The second poster shows sort of our expanding search for lead gene targets, essential gene targets in Acinetobacter and E. coli, and we show for the first time that not only do we have in vitro leads, but we demonstrate synergy with these antisense molecules and classic antibiotics, and we demonstrate that on this poster, that the 2 of them together are better than one, even in various multi-drug resistant strains of Acinetobacter or E. coli. And the third poster is one of our first forays into modulating virulence. And as a proof of principle, we started working with Burkholderia cepacia complex, which is an opportunistic pathogen that causes disease in a variety of settings, but particularly cystic fibrosis, where we try for the first time to actually modulate the formation of biofilm. And everyone knows about biofilms, this collection of organisms that can stick to various surfaces, including the lung, and are really problematic clinically for the physician because they're very difficult to treat with antibiotics, whether you're a biofilm on a tooth or on a catheter or in the lung. So we take the approach of trying to target the production of the biofilm itself as a potential strategy to deal with some of these infections. And we show in that picture that not only can we prevent the formation of biofilm in Burkhold area, but we can actually deconstruct it, an existing biofilm.
Tell it to become planktonic.
With antisense molecules. So that's a third complementary example of ways that we're thinking about using these.
So as I was speculating in the beginning, this is the brave new world, or the new era of antibiotic development, not looking at the traditional targets of cell wall and protein synthesis and, you know, anti-metabolites. This is really going after gene therapy, where you're actually targeting those genes.
Yeah, I'd like to point out that one of the significant parts of these papers, these reports, this technology that we're using to silence specific genes here is really a platform technology. And we can reduce expression of any gene with a known sequence right now. We can very quickly make new compounds that will target any gene with a known sequence. Therefore, the world is the genomes now. We're mining some of the data from these genome projects, and this technology could be applied to silencing any gene in bacteria with a known sequence. So the significance of that is that in developing a new drug, we eliminate one of the first most laborious and costly steps in drug development, that is discovering a new antibiotic or a new drug. We already have the platform technology, and within a matter of days can make a new drug. We don't have to work at discovering
You can ask the question, will this have the desired phenotypic effect on whichever microbe you may be looking at?
We can, exactly. So while we have examples of these compounds that are targeted to NDM-1, to other antibiotic resistance genes that we can silence and restore susceptibility, we also have examples of the compounds that are targeted to the essential genes like Dave mentioned, and also now to virulence factors such as those that control biofilm formation. And this is just in 3 pathogens or 4 pathogens. Potentially any pathogen could be targeted by this technology.
So you're bringing us hope instead of the normal,
I hope so.
The normal message that we typically hear are there are no new antibiotics, and we see the advocacy for 20 by 2020, the Longitude Prize, where people are begging folks to develop antibiotics, and here you're offering us a platform-based technology encouraging us to get busy.
Yes, I think so. I think that's true, and I think you know, we see an advantage to this approach, and you know, again, I'd like to point out that there's Sarepta Therapeutics is involved in this. Not all companies, you know, see that antibiotics or development of antibiotics as being potentially rewarding, but Sarepta does, and so we, in partnership with them and NIH, you know, we think there's a way forward to develop antibiotics against a great variety of bacterial pathogens. And also, I should mention too, viruses. There's active work now in developing these against a number of different viruses.
Yes, it's unfortunate that all of our enemies have nucleic acid, but it's also fortunate that they do have nucleic acid that you can target this technology to. You're watching ASM Live at ICAAC, and I'd like to invite the audience, should you have a question, please come up to the microphone and ask your question of Dr. Geller and Dr. Greenberg. Please.
I just wanted to get something clear. Does the antisense molecule itself cause the 3-log drop in bacteria, or is that when you combine it with the
No, it's not by itself. There's no effect on viability whatsoever without the standard antibiotic given concomitantly.
Okay, and how confident are you that the Antisense molecule will not have off-target effects that you're not expecting?
The evil microbiome question, that these 12 MERS are not going to wreak havoc in our gut. I guess that's target specificity, and you may speak to that.
So we haven't done the toxicology yet. There has been one report about 7 years ago that addressed that issue, and it used a slightly different compound, but similar compound, and the safety profile looked pretty good, and that was in rat, but we really haven't done extensive toxicology testing yet, but that's in the works.
I'd like you to speculate, because I know you don't know the answer to this, but when do you think that your Antisense molecule will find its way into the clinic?
Go ahead.
Well, we'll start with a lowball question. When do you anticipate you'll go phase 1 trials or things along those lines?
Well, I don't have
It's
a
a
date.
crystal ball.
That's right. Because this is sort of a platform approach, we are doing a lot of things in parallel. And so while we're looking at antibiotic resistance, we're looking at compounds against essential gene targets, we're looking at compounds against virulence gene targets, and fortunately all those things can go in parallel. So at some point, I hope in the near future, meaning within the next year or two, I believe that we will have a handful of leads that we've established as the ones that we want to move forward with further development. And I hope that we're not talking 10 years when I say we want to be doing phase 1 and advanced studies. We would like that to happen much sooner
In
than
real
that.
time.
Many of these we've already taken into animals, and so over the next year or so, some of these will probably get into more advanced animal tox level kinds of studies, and then move forward from there. So I'm not viewing this as a decade-long process. I'm hoping that the first ones are going to be much sooner than that.
Thank you.
Another question from the audience, please.
Actually, not a question. Mike Wong from Sarepta Therapeutics. I'm the Senior Director for Infectious Diseases. With regards to the question about affecting the microbiome, we know the PMOs actually don't get excreted into the gut at all. So we can actually, even given intravenously, it's primarily excreted through the urine. So you really don't have any of the microbiome effect, and really kind of saves the GI tract from any of the exposure to this agent itself. And as far as the other question, I think Dr. Geller and Dr. Greenberg answered it absolutely correctly. We're trying to move forward so we can take these into more advanced animal studies, so we can actually start looking at phase 1 human studies shortly.
Exciting times. Another question from the audience, please, and thank you for your comment.
Hi, Elizabeth Alexander from the Medicines Company. So one of the things that has plagued development of genetic-based screens for these resistance elements has been clonal diversification of resistance-conferring genes. So I'm wondering how you guys are trying to circumvent that problem, and how your KMERs are going to account for the fact that these genes are constantly mutating. Small SNPs or indels could potentially impair binding of your KMERs to the product.
Yeah. So a couple comments, and this also a little bit addresses the idea of theoretic off-target effects. So it turns out, even though these are short oligos that we're using, where you position them probably has, and we know, has a large impact on efficacy. And it turns out that by targeting the message right around the start site of the message, and we'll show that today, is very important. And it's probably important because what we're doing is inhibiting translation and preventing the ribosome from binding to its site. The nice thing about that is if you look at a genus of organisms, Acinetobacter. It turns out that around that region of the gene across species, there's very high homology. And so the same is true for many of these antibiotic resistance markers, that right at the start, there's high homology. And so our hope is, and if we've done this bioinformatically, that we actually can find targets that either cover an entire genera of organisms, or in the case of antibiotic resistance, cover a portion of the antibiotic resistance gene that isn't mutating at the same rate or under the same level of selective pressure. That's also beneficial because that cuts down on the theoretic number of off-target effects in other genomes, because by definition, it only seems to work in particular areas of the gene.
So the follow-up question would then be, have you examined the rate of outgrowing mutants when you're suppressing these targets?
Right, so that's a very important question. Bruce has published on this already. As everyone knows, resistance is a biologic trait. So the question for an antisense-based approach would be, what are ways to modulate or circumvent antibiotic resistance? And one of our grants is dealing with that very question. And you could imagine there are different ways to do that. If you thought you had sequence-based mutations, one viable approach would be to have more than one target. And I sort of think about this like I would think about tuberculosis. Perhaps you would need more than one antisense molecule. Obviously, there are ways to get resistance, and this is what Bruce's paper dealt with, about delivery, getting the molecule into the cell. And we use various peptides as delivery mechanisms right now, and it turns out that not every peptide behaves the same way. So using different peptides could be a way to circumvent this. So we're actually actively looking at mechanisms resistance. Bruce has done this in E. coli, but now we're starting to do this in some of the other pathogens that we're looking at to see if there's similar mechanisms of resistance or different, but next year. Those are ongoing.
Great question. Another question from the audience, please.
Hi, my name is Vic Arora. I'm from Grifols Therapeutics. First of all, congratulations. I think that was very exciting work, and I've been following this field for a long time. One of the key attributes, I think, is the fact that this is a platform technology, like you mentioned. So once you know how to do one, you could do multiple others. My question to you really is, if you can go after one gene, why not another one? Why did you pick the metalloproteinase? Why not a constitutional gene that would stop replication, for example?
We've done that, and there are examples on the posters that we've targeted, at least 3 or 4 different antibiotic resistance genes, and you can see we had the same effect. A reduction in viability, reduction in the MIC of the standard concomitantly added antibiotic. We also have multiple examples of essential genes that can be targeted. In fact, one of the papers we've looked at, what would you say, maybe 50 different targets perhaps or so for E. coli and Acinetobacter. And we find we do find some genes seem to be better than others, and we don't understand exactly why that is. One of our favorite genes encodes the acyl carrier protein, and that seems to be targeting that's more effective than, say, some of the other essential genes, and we don't know exactly why. We have hypotheses and some ideas, but we're just not sure why some gene targets are better than others.
So one more follow-up. So the other thing that's been really rate limiting for this technology has been just ability to push this antisense into the right cells and within cells into the right compartment, right? And clearly you're overcoming that, and that to me is really the most exciting part of it all. But when you see different bacteria, for example, do you see where you have better success or other bacteria where you have less success, or is it generally what works for one works for everything?
That's like the million-dollar question.
So, as I said, we use right now peptides, which are thought to be sort of the delivery vehicle for getting these oligos into cells. Because antisense has been around for a very long time, and people have tried this in bacteria with not a lot of success until linking these to some of these peptides. And it turns out, and the poster will show this, that not every peptide works in every pathogen, and we don't understand that, but some peptides seem to work in Burkholderia very well and wouldn't work in Acinetobacter and vice versa. So we actually have been thinking a lot about delivery into cells. We know they get into cells, but it could be to some degree peptide tropic. That actually has some advantages for us because that has, if we find groups of peptides that are effective, again, that's sort of a mitigation strategy if we were thinking about resistance and delivery. So not every gram-negative, which is mostly what we're talking about today, they're not all equal, and there are slight differences.
Thank you so much.
Thank you. You've been listening to ASM Live here at ICAAC. I'd like to thank my guests here, Dr. Bruce Geller from Oregon State University and Dr. Dave Greenberg from UT Southwest for joining us this morning discussing this exciting topic. Please join us again at noon when we discuss the emerging outbreak of Ebola in Africa. We'll have experts from CDC United States, CDC Canada, and one of the attending physicians who took care of one of the missionaries. It's been my pleasure to conduct these sessions here at ASM Live. I'm Michael Schmidt from This Week in Microbiology, and please join me and my co-host, Mocilio Schechter from Small Things Considered, Michelle Swanson from the University of Michigan, and the host of the This Week series, Vince Racaniello from Columbia University, for our next edition of TWIM, the podcast about the unseen life on our planet Earth. Thank you, everybody, and look forward to seeing you at noon.
Once you're in the BSL 4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.